Okay, children, let us discuss about yet another topic under the chapter Principles of Inheritance and Variation. The topic is non-Mendelian inheritance. Non-Mendelian inheritance, and we know that Mendel first conducted a monohybrid test cross in which he selected the character height of Pisum sativum, and the height had two opposite traits, namely the tall and dwarf, tallness and dwarfness. And in the monohybrid test cross, he observed that in the first generation, F1 generation, first filial generation, one of the characters got suppressed and only one character, one trait got expressed, and he called the expressed trait as a dominant character and the suppressed one as a recessive. Clear now? We know that the letter capital T denotes the tallness and the letter small t denotes the character dwarfness, and when both capital T and small t, what are these called? These are called as alleles. Alleles representing the character height, and when these occur together, capital T and small t, only capital T gets appeared and the small t gets disappeared, and the appeared character is known as dominant character and the disappeared or suppressed character is known as the recessive character. Clear now? So when capital T and small t occur together, what happens? Only capital T shows his presence and small t gets suppressed. So he called the expressed character, that is capital T as the dominant character and small t as the recessive character. Clear now? So in the presence of a dominant character, the recessive character gets suppressed, does not show its presence. Okay, so this type of dominance is known as complete dominance. Okay, so in Pisum sativum, what type of inheritance is observed? Complete dominance. That means when two alleles occur together, only one between this gets appeared, shows its presence, and this type of inheritance is known as complete dominance. Clear now? Complete dominance. Okay, so but some other dominance also are observed, namely incomplete dominance, codominance, multiple allelism, polygenic inheritance and pleiotropy. All these are not studied by Mendel, so these are known as a non-Mendelian inheritance. Incomplete dominance, codominance, multiple allelism, polygenic inheritance and pleiotropy. So we are going to discuss about the incomplete dominance today. So what is incomplete dominance? It is the inheritance in which the offspring shows characteristics intermediate between two extreme parental characteristics. That means the dominance in which both the characters of the parents gets mixture, the character intermediate between two parental characteristics. Clear now? In incomplete dominance, what happens? The parental characters gets mixed, blended together, and this shows what? Incomplete dominance. No dominance of one allele over the other is observed. So here what happens? Incomplete dominance is observed. What happens? If capital T and small t occur together, capital T is not dominant over the small t. So let us illustrate that with the example of the plant snapdragon or dog flower or antirrhinum. Okay, here we can see that in the picture, that is snapdragon, otherwise known as dog flower or antirrhinum species, and another plant also which you are very familiar with, known as 4 o'clock plant. 4 o'clock plant which is known as Mirabilis jalapa. Mirabilis jalapa. Commonly we call it as 4 o'clock plant. Maybe in your garden also see different colors, different colored flowers can be observed here. So why? Because this is due to incomplete dominance. Okay, so let us see what happens here. See, it produces red and white flowers. Red and white flowers. And if you represent the red by the letter capital R and white by the letter small r, and hybridization undergoes and gametes are formed from the capital R, but the gamete produced is capital R, and from the small r, gamete produced is small r, and they come together or they get fertilized and form the genotype capital R and small r. Okay, capital R and small r. And see, in the case of if it is Mendelian inheritance, what type of progeny should be observed? Red colored flower, isn't it? Red colored into white colored flower. Then if it is Mendelian inheritance, the flower form should be what? Red in color. But see here, is it red? No, it is not red, it is pink in color. What has happened here? Red and white mixed. Okay, and which color is formed here? Pink color is formed. That is why we said as a intermediate between parental characteristics. Mixture, intermediate. Okay, in between. Mixing of characters. If what color is formed by the mixing of red and white? Pink. So the F1 generation had what? Pink colored flowers. Okay, now you know the formula. Next, what is the step? Undergoing selfing or self-fertilization. So all these F1 generation were allowed to self-fertilization. Then if we cross them, see, gametes are formed. Then if we cross them, F2 to get F2 generation, but capital R small r into capital R small r. Then two types of gametes from both the parents, capital R and small r, and from other parent also capital R and small r within the circle. Okay, when these combine, say 4 types of flowers or plants with the 4 different types of flowers can be formed. See, one capital R and 2 capital R small r and 1 small r small r. Now these are the genotypes, capital R, then capital R and small r. These are the genotypes, genetic constitution. Then see the phenotype, capital R is what? Red in color, whereas capital R and small r pink in color and small r white in color. So 3 different varieties, that is red, white and pink colored flowers can be observed here. Why? Because of incomplete dominance, mixing of characters has been observed here. So you see the ratio here, phenotypic ratio is 1:2:1, that is 1 red, 2 pink and 1 white. 1:2:1, 1 red, 2 pink and 1 white. And look at the genotypic ratio, same like that of phenotypic ratio, again 1:2:1. 1 capital R and 2 capital R small r and 1 small r. So phenotypic and genotypic ratio are same in the case of incomplete dominance. So phenotype and genotype are same. Clear now? And this is asked many times for the examination, very important. And what type of dominance is observed here? incomplete dominance. Phenotypic ratio 1:2:1. 1 red, 2 pink and 1 white, and genotypic ratio 1:2:1 again, that is 1 capital R and 2 capital R and small r and 1 small r. Okay, so this means, what does this mean? This means that capital R was not completely dominant over small r. Okay, now it is not only found in a 4 o'clock plant and the example we studied. So pea plants also show some incomplete dominance in some traits. Okay, pea plants also show incomplete dominance in some traits. Okay, so I wind up here. We'll meet in the next class.